Welcome to myeyeacademy.com. I'm Professor Mahfooz from Jinnah Medical College, Pakistan. Please do give your feedback at the end to improve the contents of the website and to improve the lectures. We are going to discuss clinical physiology of eye in this lecture. First of all, what is vision? Vision is a special sense by which qualities of an object such as shape, size, luminosity and color. So if these qualities of a subject are perceived through a process in which light rays are converted into electrical signals in the retina, and then the electrical signals are conveyed to the brain via the optic nerve. So for the vision, we should have a functioning retina and the functioning optic nerve. If these two are not functioning properly, the vision process will not be completed. After defining vision, we need to learn there are four types of visual stimulation or senses, which are light sense, which is predominantly a rod function. It permits to perceive the light and allows the visibility in different lighting conditions, which means that because of the light sense, we are able to see during the day and we are able to see during the night. The next one is the form sense, which is predominantly a cone function. This form sense is the actual visual acuity that we normally measure in the clinic with the help of a Snellen's chart. The third sense is a contrast sense, which is perceiving changes in the luminance, and it is measured with a special type of a chart called contrast sensitivity chart. The fourth one is the color sense, which is perceiving colors, and we will discuss that in a bit more detail in the coming slides. All these four senses are transformed by the retina into electrical signals, and these electrical signals are transmitted to the brain via the optic nerve. So for the vision, we need to have a functioning retina and a functioning optic nerve. Out of these four senses, we will discuss in a bit more detail the visual acuity or the form sense and the color vision. Visual acuity or the form sense is measured in numbers as 6/6, 6/9, 6/12 and so on. And we measure the visual acuity with the help of the Snellen's chart, which is at the left hand of the slide. In this slide, we have got two subjects, subject 1 and subject 2. Subject 1 can see the 5 lines and is labeled to have visual acuity of 6/12, while the subject 2 has got the visual acuity of 6/6. What does this mean? This means two things. The one is that the subject 2 has got better vision as compared to subject 1, so better that the subject 2 can see a 6/12 line from 12 meters as compared to the subject 1, who can only see it from 6 meters. Another example of the measuring visual acuity is the subject 1 can see the 4 lines and has visual acuity of 6/18, while the subject 2 has got visual acuity of 6/6. What this means is that subject 2 has got better vision as compared to subject 1, so better that the subject 2 can see 6/18 line from 18 meters as compared to subject 1, who can only see it from 6 meters. The next vision sense we are going to describe is the color vision or the color sense. It is the ability to discriminate between different colors, and it is a function of the cones. In the retina, we have got 3 types of cones, which are red, green and blue. With the help of these 3 basic colors, we are able to distinguish between thousands and thousands of different colors. Color vision is tested by Ishihara chart and Farnsworth-Munsell 100 hue test. Ishihara chart is a book with the colored numbers, and you have to read the numbers to pass the test. In the Farnsworth-Munsell 100 hue test, there's a wooden box with the different colors, and you have to arrange the colors from dark to light in a sequence. Now the color vision defects are the color blindness is of 2 types, congenital and acquired. Congenital color blindness is mostly anomalous trichomet, which means that all the three cones are present but they are not functioning properly. So congenital color blinds have got all the three cones, but one or the two types of cones are not functioning properly. In the acquired color vision defects or in the acquired color blindness, there are two types of defects: the blue-yellow defects, which I have described with the help of this mnemonic bye-bye, B for blue and Y for yellow. And blue-yellow defects are in macular pathology, while the red-green defects are in optic nerve pathology. Optic nerve function is measured with 5 clinical tests. When the optic nerve is not functioning, there will be decreased visual acuity, decreased color vision in the red-green spectrum, diminished light brightness, afferent pupillary defect, and the field defects, which are usually peripheral. With the help of these 5 clinical tests, we distinguish between the optic nerve pathology and macular pathology. Visual acuity will be decreased in both. The color vision is decreased in both, but in the macular pathology it is in the blue-yellow spectrum. There will be no diminished light brightness in the macular pathology. There will be no afferent pupillary defect in the macular pathology, and the visual field defects will be mainly central as compared to the peripheral visual field defects in the nerve pathology. So these 5 clinical tests help us to distinguish between the macular and optic nerve pathology clinically. At the end, we are going to discuss the physiology of the aqueous humor. Aqueous humor is produced in the ciliary body, which is triangular in cross section, and it is mostly drained through the trabecular meshwork. After being produced in the ciliary body, the aqueous humor comes into the posterior chamber, then through the pupil comes into the anterior chamber, and then gets drained via the trabecular meshwork into the canal of Shalom, and then into the venous circulation. This route is responsible for 80% drainage of the aqueous humor. The rest of the 20% goes to the suprachoroidal space, and through the suprachoroidal space it goes to the venous circulation, and this is responsible for 20% of the drainage of the aqueous. So all the 100% is produced in the ciliary body, and all the 100% eventually ends up into the venous circulation. Now you have to remember 2 things here. The first, anterior chamber is in front of the iris and the posterior chamber is behind the iris, while the posterior segment is behind the lens. And the second thing you have to remember is all the aqueous is produced in the ciliary body and all the aqueous is drained into the venous circulation, 80% through the trabecular meshwork and 20% through the suprachoroidal space. Thank you very much for being with me at myeyeacademy.com. Thank you.